Welcome you once again to the class of drug discovery. So dear student, we were discussing about the protein modeling methods, structure modeling methods, and what are the different computational modeling methods like ab initio method, homology modeling, threading method. So these were the different methods. Now we are going to discuss about the ab initio prediction method. What is ab initio method? So I have already explained you about this. In case of low sequence similarity, we will use this method, ab initio method, ab initio modeling. What is low sequence similarity? Means during the sequence alignment. What is sequence alignment? That is comparison of the sequences. Because all the computer modeling method, protein modeling method uses the sequence identity as the criteria to model the protein. Because the only data we have input, we have that is sequence. Sequence you can obtain from the NCBI database. So if a structure is not present in the X-ray databases like PDB data bank, then you can use these methods like ab initio, threading and homology modeling methods. So for any these three methods, we have to perform the sequence alignment. Means sequence alignment of this unknown sequence with the already existing protein sequences in the databases. So we have a known protein sequence, uncharacterized protein for which we do not have structure and we do not know anything about the function of the protein. So that is our unknown protein. So in that case it is very useful. Then you will compare this unknown sequence with the already stored sequences that are already stored in the database like NCBI gene bank. You have the protein sequence. You can compare the unknown protein sequence with the already existing protein sequences. Why we will do it? To find out the suitable templates. Okay. So in case of all these three methods, the sequence is compared, the unknown sequence is compared with the already existing protein sequences. Now on the basis of protein sequence identity, you will choose the method, that which method should be used to perform modeling. Suppose sequence identity is very low. Suppose it is less than 15% or 10% also. So you can use this ab initio method. This method is suitable for that. So how we proceed that? In the ab initio prediction method, fragment library is used. There is a fragment library in which the protein structures are available. Protein structures of the different parts are available, and we also use in the ab initio prediction the global conformation space, the lattice model, discrete state models, molecular dynamics methods, preset libraries of fragment 3D models. So these three methods are used for ab initio predictions. We combine all these three. So what we do in the ab initio prediction, we have unknown sequence, then we will make the fragment of that unknown sequence. We'll cut down that. Suppose for the easy analysis we do that. Suppose there are the 10 parts of the one sequence for the unknown sequence. Now we will compare each part of sequence from the fragment 3D motif library. For the library of fragment 3D motif, it will be compared to that. Okay, preset libraries of fragment 3D motifs. So suppose for unknown sequence it has a 10 fragment. We have made the 10 fragment. Then each fragment will be searched with the fragment structure of the 3D motifs. I mean that is already saved in the library. So that is very important step. If we find the structures, short structure, then it is very useful. Okay. Molecular dynamics methods are also applied That is also very important, molecular dynamics methods. So the step that we have discussed, that the searching of the structure in the fragment library, that is similar to the homology modeling step. We will discuss it, okay? Because we are copying the structure from the library fragment, and then we will combine those. So searching of the fragment structure from the library, that is easy. So they will search the similar structure, and then we can combine the structure of the 10 parts. So again, combining the structure of the protein fragment, that is a difficult process. So in that we have to apply the algorithm. Computer has to apply the algorithm. So it is done. This step is done using the computational algorithms, so that the different structural part can be combined according to the conformation. Because the protein structure conformation, it shall be similar to the native protein. Like, I mean, it should not violate the rule of the native protein structure. Although we do not know anything about the non-protein structure, but we have to make the realistic model, means that is close to the reality. Okay, so how it is, how we can determine, how we can predict this is the close to the reality or not, this will be the native structure or not, this will be the probable the protein model that we have created using these 10 different parts, and we have copied the structure from the library. So how this is, in which way the order is important, in which way we are combining the different structure. So that is very important. The model should close to the native structure. It should be probable native structure. How we are going to determine that is. So different algorithms are used for that step. So it is done on the basis of, by the comparison of the structure. Okay, the non and different fragment structures are compared. So distance between the atoms is measured in terms of RMSD, that is called root mean square deviation. Root mean square deviation. RMS is calculated, that is the coordinate root mean square. So how many atoms are involved, how many coordinates are involved, and that is divided by the total number of atoms. Okay, उसको हम calculate करते हैं. ये मैं I will tell you in the separate lecture what is root mean square deviation. Now the RMS is the measure of deviation, means it measures the distance between the model structure and already present model. So if the CRMS is low, then it is thought to be considered good. It should be low. It should be below 2 angstrom. The value of the RMS should be below the 2 angstrom. Then it is considered good. Okay. So here the lattice model, discrete state model, this or molecular dynamics, these things are also written. Sometimes we apply the lattice model. So we make the lattice models according to the hydrogen bond conformations. Like in the space, we draw the structure of the sequence on the basis of the like bonding patterns of the amino acids, whether this can make the hydrogen bond or not, whether the amino acid can make the hydrogen bond or not. According to that, we produce the lattice model. So in the right hand side, you can see this is the lattice model which is created in the space. Okay. Molecular dynamics methods. So what is this? So are they the combination of these methods? So sometimes no part of structure is matched. We have divided the sequence into the multiple part, but no part of the sequence is matching with the already existing protein sequence. Then what will you do? Means, or suppose for the already existing protein, if suppose there is no structure in the PDB data bank, because for these methods to apply, we should have the structure in the protein data bank, then we model the sequence. Suppose there is no structure present, but ab initio method is useful in this case if there is no structure of the protein, that is you have compared that protein sequence with already stored sequences. Suppose using the blast method, generally blast method is used. So from the NCBI database sequence, protein sequence, a protein database, you can obtain the sequence. Suppose there is no structure that is present in the PDB data bank, then also this method is useful, because it also uses the molecular dynamics methods and lattice model also, that is based on the properties of amino acid, atomic properties. So lattice model also use the amino acid properties, and molecular dynamics also that use the atomic properties. What are these? Electron, proton. So what is the molecular dynamics? It doesn't take the sequence into account. It is not based on the sequence, and we are not copying the structure. Okay, we are not copying the structure. So this is the picking native conformation with energy function. So energy function is important, means how we are going to create the complete model. It should be the conformation with the minimum energy. Energy is also the one criteria. If the model protein has the minimum energy, okay, then the protein model is good. You can compare it. If the three models that are they have the minimum energy, and which one has the lowest energy, that is a good model actually. On the basis of energy you can predict. So it based on the energy function, based on the solvation model also, how protein interact with each other, interacts with water, and pair interaction between amino acid. Actually folding occur in the aqueous environment. In our body we have the aqueous environment, and protein get folded in that environment, in aqueous environment. Hydrophobic residue come in the the R group phase to the core group. So in the core region we have the more hydrophobic residue, the non-polar groups, okay, and polar groups and polar amino acid are found on the surface of the protein. So that is it. So pair interaction between amino acids are important for this. Then only the energy will become negative. So in our body it happens. Protein has the conformation, spontaneous conformation. If it has the minimum energy, so minimum conformation energy. So that is a favorable conformation. And in the reality it happen, and structure will have the native structure or the realistic model. Now what we do in the molecular dynamics, it is means in the molecular dynamics we analyze the time dependent behavior of the molecules, how it behave over a time period like T1, T2, T3. So in the molecular dynamics we use to analyze that. There can be various stages of the protein, so protein can change the structure also. So if you want to analyze the protein dynamics, you want to see the protein dynamics, then this method is suitable also. So we carry out the simulation, that is called simulation with the mathematical modeling, that is done on the basis of atomic properties. What is the molecular dynamics? It is done on the basis of atomic properties. What is atomic properties? What is the property of atom? Can you tell me? Like position and momentum. Position and momentum. So position is important. So how you are going to observe the energy changes? Suppose anything you have kept in your room in the morning on your table, and in the afternoon when you are coming to home and you don't see that thing on the table, then you know that that thing has been moved from the table to the another place. So that is the displacement of the atom. That means somebody has applied the energy. So without energy there is no change. if somebody has moved from that particular thing on the table from one place to another, then you will analyze that somebody has come in my room. So somebody has applied the energy. So energy is important, and if it is changed, if it has a slight change, that is spontaneous. That is spontaneous change. One can do it without using any energy, means minimum energy. So any favorable change has a minimum energy. Okay. So energy is the function of, how can you define the energy? Energy is the function of coordinate also, because the thing that you have kept on your table, that will have the different coordinate, and in the afternoon or evening, you will see that the structure will have the different coordinate, means x, y, z coordinate. So that is important. So energy is a function of coordinate like x, y, z. So in the so for one thing you can say there is a coordinate x1, y1, z1. These are the three coordinate. And for the another time you can see, for that at the time t2 you can see like there can be coordinate x2, y2 and z2. If there is a change, so energy will be changed. Energy is changed. So this happens. So that thing we analyze in the molecular dynamics using the atomic properties, because positions of atom or the momenta you cannot see. You can see the water is moving in the bucket, but you are not seeing the individual atoms. So molecular dynamics takes that property of atom into account. It is based on the position and momenta. So what we do in the simulation, we initialize any reaction. So we use the mathematical modeling method, because the large number of molecules, large number of atoms are involved here. So we calculate, we initialize the atoms using the position and momenta, then we see the change. We provide the different conditions within the computer. It happens. Simulation we carry out in the computer. So you can provide the different temperature also, T1, T2, T3, T4. So there may be the various temperature also, 25, 26, 27, 28 degree temperature you can provide for the protein conformational changes, and you can analyze the what are the changes that are occurring. Okay, the protein are going through. There are the so many conformation changes. When we take the drug, there are the conformation changes in the protein. So you can analyze those kind of process using the simulation method, because in the simulation we need the protein structure file like PDB structure. We should have structure. No, in the molecular dynamics no sequence is used. It is not sequence based method. In this, structure is used. We should have the structure file. We should have the structure file. Then we can apply. So there is a combination of the method that is used, sequence based molecular dynamics. So atomic properties are taken into the account, and we make the equation like kinetic energy. What is important for the movement? Potential energy, kinetic energy. We are shaking the water in the bucket with our hand. We are shaking that water. That means we are applying the kinetic energy. Okay, and you can set the ball at the top of the slope. That means you have stored potential energy in that. Then that potential energy, then you leave that ball, so it will come down towards the bottom spontaneously, because you have there is a potential energy in the ball if it is kept on the top. So but kinetic is important for the dynamics. If you want to observe the changes in the proteins conformation, then kinetic energy concept is important. Suppose the water is moving in the bucket and you want to see that in the computer how it is moving, how it is making hydrogen bond, because in the liquid water hydrogen bonds are constantly made and broken down. So we can analyze that water simulation using molecular dynamics methods. It is not so molecular dynamics is applied to analyze the time dependent behavior of the atoms of the molecules. So it is based on the atomic properties like position and momentum, because energy is the function of the coordinate. If the molecules are moving, it will show changes in the coordinate position and momentum. So in the simulation we initialize the atomic position and velocities, means and the momentum, then we see the changes. So in the computer we give the conditions like temperatures 25, 26, 27 and 28. Then we can see the protein conformational change. We can observe the changes in their coordinates. Okay, and we can predict the different conformation of the same protein over a different condition, like on condition T1, T2 and T3, so T4. So at different timings you can compare the protein conformational changes. So molecular dynamic methods is also suitable for the drug metabolism also, to study the drug metabolism, like at the various stages how it is showing the conformation, how the protein is going under conformational changes. So drug and protein conformation changes. When we take the drug in our body, it shows the changes, drug and proteins also. So we can analyze drug kinetics using the molecular dynamic simulation method. That is very important. And how we do it? So at the various condition, various stages, we can analyze the protein conformational changes. So this is very important method. So ab initio prediction use these methods in combination. It also use the fragment libraries. It also use the molecular dynamics methods. So molecular dynamics method is important because we can see the protein dynamics. We can analyze the protein dynamics, how the protein behave dynamically and how it shows the conformational changes when drug interact with the protein. So that is very important. So this is based on the mathematical modeling method. Sequence is not taken into the account. For the molecular dynamics simulation, we need the structure. So in the molecular dynamics we use the statistical mechanics also. So there are the two types of properties. One is called microscopic properties, another one is macroscopic properties. So relationship between the microscopic properties and macroscopic properties is made via statistical mechanics. So statistical mechanics is one branch because we use the statistics when we have the large number of data. So we have the large number of molecules, large number of atoms, and we are simulating that. Suppose the liquid water in the bucket and we are simulating that. So there is a large number of, even you don't know how many molecules of water in the bucket. Okay, so you simulate that model and you can see the changes. You can see in the form of the movie. So you can analyze the behavior of the molecule at various time stages using the molecular dynamics. So in the ab initio prediction, we use these different types of methods in the combination. Solution model is important. If the protein interacts with the water, then it is called solution model. So then the drug can interact with the protein. So that is called solvated model. So energy concept is important here. The reaction becomes spontaneous if there is a free energy. So delta G should be negative for any change, and for delta G should be negative, delta H or enthalpy change, it should be more negative. So that is there are other secondary structure methods also, that is called local homology, fragment library. I have already told you about the fragment libraries. What are fragment libraries? Means that are the proteins structure are saved in the fragments, like fold structure, similar motifs. Motifs are important. So motif is important here. For the ab initio prediction, the motif structures are important because in the library we have the 3D structure of the 3D motifs, fragment 3D motifs. Okay. Local homology means when we have the different part of the sequence share the homology. So that is called local homology, we can say. Okay, we will discuss it. If you have any question, you can ask. Now the lecture is over. So although we have, so in the another part we will discuss lattice string folding methods, forces hydrophobic. So here you can see this is the lattice model. Okay, so we can make the how many hydrogen bonds are possible among the atoms. You can draw it on your page, on your notebook also. Okay, so these are some examples. We were talking about the lattice string folding. These are the examples, and this is the software that is used. So software we are going to discuss in the next class. This is the software Rosetta that is based on the ab initio method. Okay, so Rosetta is the one software online tool that you can use for the ab initio modeling. Okay, Caspi also. So here you can see these are the examples. These are the examples. Structures are shown here. Okay. Okay, so these are all examples that you can see. So this type of structure we obtain using the ab initio modeling methods. Okay, this is the model 4 and this is close to the native. On the right hand side a native structure is shown and the model 4. So this is similar to this. This is a native structure for target 56 and this is the model 5. Is target 74 and model 2. So these are the examples of that. Now we have discussed the ab initio. Today we have discussed about the ab initio modeling method in the detail. So here you can see the result, how the protein is modeled. So these are the different model, model 4, model 5, model 2, and that is model using the target 77 iterative structure. So target 56, target 74. So these are the target structures, like you can call it a template. Okay, so Rosetta method is important. Rosetta you can use for modeling the structure if you want to use the ab initio modeling methods. Okay, okay, now do you have any? Okay, so these are all. Are you all able to see? These are all the structure that we get after modeling. Okay, now the lecture is over.